Humans share over 99% of their DNA. That means that if you looked in any two people and you looked at their DNA sequences, 99% of its sequence would be exactly the same. And yet there's tremendous variation in phenotypes in the human population. Similarly, chimpanzees and humans, as two different species, still share over 98% of their DNA. Yet again, their phenotypes are quite different and they're separate species. In order to understand how this can be possible, and to better understand the process of cell specialization, we're going to take a close look at eukaryotic gene regulation. And we're going to focus on mechanisms that help control gene expression, particularly at the transcriptional level. So a quick review of gene expression. Remember, gene expression is turning on or off genes, and it consists of two major steps. To get a gene into a protein, we first have to transcribe the DNA into mRNA. That's the first stage of gene expression. Then we've got to translate the mRNA into a polypeptide, which makes up a protein, which makes up a trait. In this particular video, we're going to focus again on mechanisms of transcriptional control. So the first method is DNA packing into chromosomes. If you take a look at a chromosome, it's actually quite complex at the level of DNA. So let's start over here. Here's DNA, our double helix. In order to get DNA to fit nice and neatly inside the nucleus, this DNA is going to wind around histone proteins, like this, and it looks like beads on a string. The winding of DNA around the histone proteins forms a structure called a nucleosome. The nucleosomes then wind into a bigger helix, and then that helix winds into a supercoil, and then the supercoil loops and folds, and that's actually what makes up a chromosome. So a simple picture of a chromosome may lead you to think that the structure is pretty simple, but there's multiple levels of DNA packing. And this is important to transcription because in order for RNA polymerase to transcribe DNA, the DNA has got to be unpacked. If the DNA is packed up like this, RNA polymerase can't access the gene and it can't transcribe the DNA into mRNA. Another method of transcriptional regulation is something we're pretty familiar with, transcription factors. Here we've got a DNA strand. The gene is right here. This is the part of the DNA that will actually code for a protein. Before the gene is a sequence of DNA called the promoter. We know that the RNA polymerase has to bind to the promoter in order to start transcription. But then, further upstream from the DNA promoter and gene are these sequences called enhancers. Enhancers will get bound by transcription factors. And the job of transcription factors is to help RNA polymerase start transcription. So here's what it looks like if the gene is going to get transcribed. Here we can see that the transcription factors are bound to the enhancer regions, and in doing so, they have helped RNA polymerase bind to the promoter. Now RNA polymerase can start transcribing the gene. So to sum that up, RNA polymerase is the one transcribing DNA into RNA. The promoter is this green region that the RNA polymerase has to bind, and the promoter is DNA. The enhancer is another area of DNA, often quite far away from the gene, and that's what the transcription factors bind to. And the transcription factors are these purple and green molecules. And it's important to note, you're going to want to add this to your annotation, that transcription factors can activate or repress transcription. In this particular diagram, we are seeing activators because they're helping activate the RNA polymerase. But there are other types that will repress RNA polymerase's activity and turn genes off. Another type of control is our alternative splicing, and we're familiar with this as well. Remember that in a DNA gene, not all of the sequences will end up in the final RNA. So the DNA gets transcribed into RNA, but only the exons, 1, 2, 3, 4, and 5, end up in the final RNA molecule. The introns, these non-coding regions, get cut out and discarded. And the cool thing about splicing is that you can splice the exons together in different ways. For this mRNA molecule, we've spliced together 1, 2, 3, and 5 to get a particular mRNA molecule. In this one, we've spliced together 1, 2, 4, and 5. So the cool thing about this is with one DNA sequence, we can get multiple RNAs, which means multiple proteins. And this helps explain some of the variation in phenotypes that we see within humans or with humans compared to chimpanzees. Because even if two different people have the same DNA sequence, you might get different traits depending on how the RNA gets spliced together. Another method of controlling transcription happens slightly after transcription, in fact. Here's our mRNA molecule. You can see it's got its cap. But eventually the mRNA molecule is going to get broken down or degraded. And when that happens is key, because the lifespan of mRNA determines how much protein is made. Eventually, the mRNA will get broken down by enzymes and RNA, but depending on how long it takes to get broken down, the mRNA molecule could last a few hours or many, many weeks. And the longer it lasts, the more protein is going to be made. So if a cell needs a lot of a particular type of protein, it won't degrade the mRNA right away. If a cell only needs a little bit of protein or the environment changes and it doesn't need that protein anymore, it can respond by breaking down the mRNA into little pieces so that we don't get any more protein. And there's a specific mechanism involved in degrading mRNA. It's called RNA interference. And this is a hot topic in science right now. Remember that only 2% of the human genome consists of genes that code for proteins. So you've got 98% of your DNA that's not producing proteins, that's not contributing directly to traits. For a long time, scientists just thought that 98% was junk, but it turns out that it's not really junk. In fact, some of it is transcribed into tiny little RNA molecules called microRNAs. And here's what those microRNAs do. A microRNA, here's this short little RNA, will get together with some proteins and it will bind to an mRNA molecule. And once it binds to that mRNA molecule, a couple things might happen. One, it might get degraded, thereby stopping translation, or it might block the ribosome from translating the mRNA molecule. Either way, the result is the same. We have stopped translation of the mRNA into a protein. So this is a major mechanism for controlling gene expression. And this also contributes to variation among species. And that's all for today. Please make sure your notes have all the key components and don't forget to do the poll.